DNA sequencing allows us to do so many things. Because think back to when we talked about the history of DNA and having first deduced what DNA was and then trying to determine the structure. And then after we determined the structure, we just had to investigate to find everything that we could about it. Well, now that we can sequence DNA, we can sequence genomes. And if we sequence genomes, then we can annotate those genomes to point out the parts that are coding sequences. So what's introns, what's exons, where are the splice sites, where's the termination occurring, where's the start site occurring, where are the activators binding, where's the insulators. Like, we can figure out all of those things, all of the regulatory elements and everything that a gene contains. And so we can start to compare genomes of humans to genomes of zebrafish or pandas, or even from human to human. And so, being able to sequence the DNA is really opening up a lot more doors for us. Now, this first came to be in the 1970s, and around that time there were two different groups who did it. Now, Sanger he has went on to win the Nobel Prize. His sequence was used to sequence the human genome that we completed in 2001. It made the cover of Science magazines and New York Times alike. And the idea is that, you extract DNA from a person and you can tell them the entire sequence of all of their genetic material. So what is the order of A, T, C, and G? So how they do it? Well, Sanger, the one that we're going to discuss, uses this dideoxy sequencing, right? What is that? Well, it sounds familiar to you because you've heard of deoxy, right? Deoxyribose, which makes up DNA. So what is dideoxyribose? So he took into account that, so here in C is ribonucleic acid found in RNA, and you'll notice that there are these two hydroxy groups, right? Well, to make deoxyribose, deoxy take away an oxygen. So deoxyribose has the removal of an oxygen, and dideoxy has the removal of two oxygens. So by removing those two oxygens, then it doesn't allow the sugar phosphate backbone to continue. So when a dideoxynucleotide is inserted, it causes this chain termination. Now, what does that mean? That means that you're going to have sequences of varying lengths, right? Each one ends at a different point. And what we do with that, because each of these dideoxys are labeled now we can tell what color ended a chain, right? So what fell at the end of it. So when you put it together, Sanger had to go back to those gene cloning techniques that we talked about. And so he has target DNA, small sequences of DNA that are put in these recombinant vectors, and then he's throwing at them the fluorescently labeled dideoxynucleotides and then normal deoxynucleotides so that some sequence or some complementary base pairing can occur. But every time a dideoxy is inserted, it fluoresces and it causes chain termination. So you get DNA of varying lengths, right? And now you take that DNA of varying lengths and you run it on a gel, and then the order in which you see, so this is called the ladder sequencing because you can essentially read the order from the gel. But to help automate this, then there are computer programs that picks up the different fluorescence and tells you how much you have, and that also gives you the sequence, okay? So this is the Sanger method, and it takes into account the use of dideoxyribonucleic acid. Now you have to think about what are the implications since we can essentially sequence the human genome and we can sequence genomes of different organisms. What can we do with it? So when we first sequenced the human genome over 20 years ago now, that was monumentous, okay? That was a landmark discovery. It hadn't been done before and everyone was excited. And then that begged the question, like, when will we get to the so-called $1,000 genome? So in the beginning, it costs a lot of money. But the idea is to get to this $1,000 genome where everyone can have their genome sequence. And essentially, it will become part of your medical records. Well, what are the pros and cons of that? Okay, so if you can sequence a genome and you can point out the likelihood that people would have a certain disorder or be susceptible to something, like how does that affect employment, insurance, everything? And so that's what you have to think about. Like, yeah, it's great. We can sequence the human genome. We can ask some basic biological questions, but what medical and health implications does that then draw? Because on the flip side, then you have to think about, this would also help us to figure out how people will react to medicine. So we could definitely see personalized medicine being an extension from this. Now, from all of this thing, there's the Gina Act, and that's looking at, you can't discriminate against someone based on their genetics. And so, put all these things into account, and is it worth it? Should we move forward with this technology, or should we stop it in its tracks?